research laboratory. You can see it's very spacious. We have a lot of space, which is unusual for a lot of research. Usually the labs go up higher and higher to stack things up. We're pretty high here, but not that high. The air in here is the cleanest air in Bradley University. Everything is absolutely filtered because a lot of what we're doing requires growing cells and keeping them alive and happy. And yeast, mold, bacteria spores cause a big problem. So there's filtration on all the air coming in this laboratory. It's very clean as far as the air quality.
I'll close the door.
And, well, that's true.
Not anymore.
That's right. But primarily we do a lot of cell culture, and we culture a lot of different cells in this laboratory. We culture brain cells, absolutely critical to learn how to culture brain cells. So that's a very, in fact, Swathy is an expert on culturing brain cells in the laboratory. We need brain cells because that's what we want to turn the stem cells into. So we've got to be very good at growing brain cells, neurons. We also have culture loads of stem cells, lots of them. We do bone marrow stem cells, we do cord blood stem cells, we do special subtypes of those. There's a very primitive type of cell called a CD34 cell that's found, a very rare stem cell. It's found in some cord blood, it's also found in bone marrow, and we are very good at isolating that and growing it. But it took us a long time to grow these cells. We killed cells for about 3 months before we actually did anything. And now we've learned how to freeze them in a very special way, thaw them, and keep them alive. And those are the cells that we're going to be working with, the new cells, to change them into neurons. So we've changed from bone marrow mesenchymal cells to these special CD34s. So we're very excited about those. And by the way, we get those from cord blood, a lot of those from cord blood. So.
And learned
All right.
a lot of your cord.
And I would, since Lindy and Steve are here, Swathy and Amanda, they are the ones who set up. Swathy is in charge of shipping out the collection kits. This is Lindy and Steve. That's where we've gotten, they set a program up, and thanks to them, we are getting cord blood from all over the country.
All right. Got two of my grandkids.
It's just,
They're having grandbaby in March.
We have Dr. Cady. You've got some cord blood from Cheryl and Chad's
thank you very
children. When you were on the show, Chad had called in.
Oh, that's right. You had called while we were. That's right. We called in.
You got a couple of my grandkids' blood.
Your grandkids.
Hopefully they did some good.
It did some very good. We used those cells and right away to work with that.
So worry about my
Here's my second-year student who is hiding over here.
Hi.
This is Carrie Hauser. Carrie has
Carrie.
designed and worked with the CD34 cells. She's killed cells for 3 months. We got really upset with it. But we had to learn.
Got to be good at something.
That's right. She's now very good at keeping them alive and freezing.
All right.
She's an expert on that, and she's come through that very difficult process of having things not work for about 2-3 months, and we're finally very good at it. So Carrie's perfected that technique.
Excellent.
And she's a second-year student. So.
Keep up the good work, Carrie.
The lab we obviously we need a lot of high-tech equipment in the lab, and for us, the big hood over there that you can hear making background noise way back there in the corner, over there, that's where we work with all the stem cells, all of our cells. Inside that glass hood is a completely sterile environment. The airflow is internal, and it's all filtered, so it's ultra pure, and there are no yeast-borne bacteria.
You want to be on video? There you are.
It's pretty clean, but now it's clean.
I gotta steal stem
So
cells.
all of our stem cells.
Looks like a little incubator over there.
So the students are wearing lab coats and gloves, not only to protect themselves, but also protect the stem cells also. The incubators over there, Amanda, can you open the incubator over there? We have very special incubators, by the way, for almost all laboratories. This is where we store the stem cells, the cancer cells. These are special because most laboratories just control carbon dioxide gas. We're very different. We control both oxygen and carbon. It's called triple gas because we use nitrogen, oxygen, and carbon dioxide.
Now, what do you do with the incubator? What do they do with the incubator?
All our cells stay in it, so this keeps it at the right temperature, the right gas levels.
Do you keep track of the blood and the cells you get the donations from and what you're working on, or?
We keep track of some, but we don't have names necessarily
Right.
where they're from.
With my 2 grandkids, I just sent within a month and a half, I sent two blood donations.
Oh, in the last couple months?
English and last name on the other one would be Durham.
Yeah, I actually remember that.
Yeah, good.
I processed that, yeah.
Cool.
Wouldn't it be cool if she said, like, I don't know?
Yeah.
You should do that just to freak people out.
Well, my son, I'm going to share this video to my son and
Yeah, that'd be
my
cool.
other
The other
stepson.
thing I want to show
Show
you
where they ended up
is,
at.
Yeah, that's a good idea.
the microscope. It's a very special microscope. Very expensive, of course. I mean, everything's expensive in here, but this is a special microscope. It allows us to essentially look at living cells. A regular microscope like that, you stain the cells that are usually dead. This is for looking at living cells. And we have a very fancy camera on it, digital camera that shows it. But if you look at it, something unusual, the whole thing basically rides on a cushion of air. If you don't believe me. And the reason for that is because what we do in here is we listen to one cell at a time electrically. One single cell. A lot of people don't, right, Swathi? We do that.
Yes.
One single cell. So any vibration would tear the cell apart. Why don't you, can you hold up the electrode,
That's one thing
Swathi?
that I find interesting.
We use a special glass
Yeah.
tube. We call it an
They use a
electrode.
PC.
It looks like a glass needle
Whatever,
to
the Mac,
touch one brain cell.
I'm not Mac PC or
And
Linux.
listen
I
to
like
it
them all,
electrically.
but
Maybe I'll send this video
And
into
the idea, it's
PC
a
and
simple idea.
they
Because
can make this
if it
about
sounds
Mac.
electrically
They never
like a
made it to the
neuron,
lab.
that's
it's
good.
changed. That stem cells
good- that's
change
good one.
into a neuron. And there are very few laboratories doing that. And that's where we feel we have a great advantage. Swathi is learning that technique. And it's a difficult technique, obviously. We have to use all these special equipment to move it. That instrument and the one with the little microscope on it, that's all they do is make that little glass needle.
Question, we noticed that you were using PC instead of Macintosh.
Yep.
PC is good, right?
Well, we use PCs to acquire data
Right?
and to work with data, but I use a Mac for my personal, so I've got to know
Okay.
both Mac and PC.
Oh, I was going to make a PC commercial out of this.
You have to hide my Mac then if you do
Okay.
that.
You notice the Mac is in the corner.
That is. I think I know his preference already.
Have
so.
we used Linux at all?
Yeah.
And then the other thing we do in the laboratory is we also do something called immunohistochemistry. And it's a big fancy name, but all it means is we're looking for proteins that are only found in neurons. That way, or brain cells. That way, if we change a stem cell into a neuron, it should also have only those proteins that are found in neurons. So if we've converted it all the way into a neuron, it should have proteins only found like in neurons. So we do that technique. We use lots of antibodies, fluorescent colors, and things of that sort to do that. But this technique, electrophysiology, electrically listen to it, that's where that's a very exciting technique. It's very exciting. And I think Swathi is this neuron.
This is our cortex that we've cultured for
Great.
a while.
Carrie, can you turn the lights off for a minute? If you come around and take a look, Carrie is going to show you some of the neurons that we grow. And she can show you and brag about how healthy her neurons are.
These are the ones that we just cultured. It's been about, they've been going for like 3 or 4 weeks.
These are living neurons.
So they're living, and these are the ones that we actually use to do the patch clamping where we touch it with a little tiny glass tube that we just saw you guys. And this is from the cortex, which is the outer region of the brain. And so we collect it from 2 areas when we culture them. We do the hippocampus, which is more in the inner area, in the middle, right? Almost. And then we do the outer area, which is the cortex. And so this is one of them. And the way you can tell that they're healthy is that they're like kind of, because it's a phase contrast, you can see stuff, like you can know that if it's 3D or if it's just flat. And the way you can say that it's kind of, they have that little halo around them, that glow. And that's basically what it was.
She's got a lot of healthy. We look at that little halo light, a ring around the cells. That's a sign they're very healthy. How long have these been in culture?
These have been for 3 weeks, almost.
These are 3 weeks.
This isn't top secret, right?
No,
no.
it's fine. Go for it.
But any other lab that I have a colleague who's also culturing these, he only keeps them alive, he can only keep them alive for 1 week.
Video.
So ours are doing very well.
Perfect.
So we're very good at neuron culture.
Get everything
brain
you
cell
can. We can set up our own lab.
So we feed them every 4 days. We change their media and the stuff that they live
This is
in.
exciting
So
stuff.
we keep them alive. We keep them happy. They're like our babies. At least they're my babies.
She's very attached to them.
So yeah.
Everybody can come and take a look at it on the screen. If you can't see, just come up, move in. There's plenty of room. You can take a look on the screen and see what they look like, or you can look through the microscope if you'd like to. And these are what normal, healthy brain cells look like. The other thing I'd like to show all of you is I'd like to show you some stem cells today. If you're interested in seeing,
Sounds good.
we'd like to show you some stem cells, what they really look like, living stem cells.
I'm gonna sign
And I
off
think
now.
Amanda can do that. She's got some. You're growing.
What's the magnification of the neurons?
Right now it's 20.
I'm gonna go ahead and
About
take
20
some
times.
photos of that.
Wow, they're bigger than I thought.
20 times, yeah.